I'm an environmental microbiologist, not a medical microbiologist. So what I'm really interested in is what happens to bacteria out there in the environment. But I also have a medical aspect to my research, because what I want to find out is what pathogenic bacteria are doing, the ones that can infect you, what they're doing out there in the environment before they get inside you. But what I really want to know is how are the bacteria out in the environment responding to the environmental conditions, and does that prepare them for infection, or does all the preparation occur once you've swallowed them and they get into your gut? A modern society has to be able to depend on its water supply. If you don't have fresh water, if you can't count on the water that's coming out of your tap, then you're always wondering about, is this glass of water going to get me sick? If I brush my teeth with this water, am I going to come down with an illness, gastroenteritis? Am I going to start throwing up? Am I going to get diarrhea? That's a very bad place to be for a society. Typically, Cryptosporidium gets into us through water supplies, through our drinking water. It only rarely comes in through food. So Cryptosporidium will come in through a glass of water that you drink, and that's what happened back in the 1993 Cryptosporidium outbreak. It got into our water supply.
Okay, we're just going to go over there to the falls, right alongside there, cast it, drag it back in.
Okay.
The question is the gene that you're looking at functional in that sort of system that you're studying? If they are turned on, that means they are ready to infect you. So the question is, are those genes turned on, ready to infect you, or turned off, not very infectious at all? So what we do is we genetically fuse two different types of genes together. The gene that we're interested in and another gene that we refer to as a bioreporter or a bioindicator gene. And what that does is provides us a signal that we can easily see and measure. The end result of all this work will be to understand how microorganisms prepare themselves for infection even while they're out there in the environment. That's one area that is overlooked quite a lot. Usually we're only concerned with what happens to bacteria after they get inside of you and they start the infection process. But I think we can back up our knowledge about what the pathogenic bacteria are doing all the way into the environment and perhaps defeat them there, or at least cripple them more there so they're not as infectious by the time they reach us.